The molecule is delivering amino acids to ribosome during translation. What is that? Transfer RNA. Question 20. The portion of the tRNA that binds to the codon of the mRNA is called what? Anticodon. In ribosomes can attach to the mRNA and begin translation even though transcription has not been completed. Which one? Same thing. Prokaryotes, is that right? Okay. The reaction sequentially links adjacent amino acids in the growing polypeptide. What did this call it? Peptidation? Transpeptidation? What did this call? Transpeptidation, is that right? Okay. We did this already. Okay. What is the biological function of DNA polymerase 1 and 3? Very simple. You should know the answer. DNA polymerase 1 DNA polymerase 3. What's the function? Okay. Polymerization. Okay, that's no question. 5' to 3'. What's the function of that guy? Hoof reading, is that right? Because 3' to 5' exonucleus. How about that one? Of course, polymerization. What else? Do you remember small sequence in Okazaki segment? Once the RNA is gone, is that right? Once the RNA primer cut is coming. What is the Okazaki fragment? Yeah. In the lagging beds, because there's no 3' hydroxy bonds, is that right? So they have to form those small pieces of the DNA sequence, leaded by RNA primer. That is called Okazaki fragments. Okay, or whatever, you can use your own language to describe that. That's okay. So all the others we talk about. So we get this one down, we're going to talk about the others. Last one. There is some more questions for you to get practice. Okay, we're going to look for this. Coenzyme. Very small proteins. Of course, it's true. It's not correct. Coenzyme is non-protein segments. You don't have to read it. Or specific for each type of reaction characterized. Not really. Never bind to reactants when attached to an enzyme to activate site. No. Or small molecules derived from vitamins. So E is the best answer. This we already mentioned. Sulfur drug, the answer is C. This is what we mentioned, fermentation. Okay, here we're going to look. The purple sulfur and the green sulfur bacteria obtain energy from growth and aerobic respiration conditions by what? Okay, this question, a little bit difficult. So what do you have to look? So you have to go back to look at the slides when we talk about E. That's right. What is that called? The sulfur monoids and the thermal protease. Right here. That's sulfur drug. Okay? So what happened? Reduction SO4 to H2S. Okay, so the answer is E. Best definition of fermentation we already mentioned. Is that right? What's the best definition? Yeah, organic molecule as a final electron accept C. Okay, this is the one. At the bottom of a swamp, you may find the black deposits produced by the metabolism of disulfur vibro. This bacteria is using what as its terminal electron acceptor? Is that C? Disulfur vibro, what is that? It's a vibro morphology disulfur using sulfate. So, this typically tells you, what's that? Typically tells you it is anaerobic respiration. So, what is the terminal electron acceptor? Is the chemicals other than oxygen. So, what is that? Then H2S. This is similar to H2O, just to change S to O. Okay, so it's C. You don't have to do that. That one, the answer could be coupled. So I don't talk about that anymore. Which of the following reaction takes place outside of the bacterial cell? Glucose to fructose with 6 diphosphates inside the cell. Pyruvic acid to lactic acid inside the cell. Conversion polymers to monomers inside. Substrate level phosphorylation inside. Removal of electron and proton from organic molecules by dehydrogenase. Of course, that happens outside of the cell. Is that right? That's part of the potential energy for chemosmosis. So the answer is D. Again, you go back to study chemosmosis. Which of the following is the best definition for anaerobic respiration? Oxidation of a carbohydrate, inorganic molecules excluding oxygen serve as the final electron acceptor. Again, go back to that. So look at here, at least 5 or 6 questions talk about definition of anaerobic, aerobic respiration, and the fermentation. Just combine them to study it. The conversion of phosphophenols gases to pyruvic acid is what? Release carbon dioxide. Involves the conversion of ADP to and the ATP. Yes. Involves substrate level phosphorylation. Yes. So what's the answer? B and C. There's no released carbon dioxide. You should not have a problem for this one because I gave you extra points. I asked you to study. Long time ago, this is going to be a problem because you don't know the cycle. If that happens here, is that right? Phosphophenols becomes pyruvates. Substrate level phosphorylation, no carbon dioxide release. Carbon dioxide release, what happens is citric acid cycle. That's related to electron transport chain most of the time. Okay, this is not. So the answer is substrate level phosphorylation happens, and it also happened is the conversion of ADP become ATP. This has become an easy question because I asked you to study for the extra points. Otherwise, this is difficult. That means you have to know the last step of the glycolysis. Question number 11. How does fermentation of glucose differ from the oxidation of glucose? What's the difference? Fermentation of glucose is anaerobic. Oxidation of glucose can be aerobic and anaerobic. Yes, the answer is A. It's correct. Fermentation produces less ATP per glucose molecules than oxidation does. Yes, the answer is correct. Glucose is fermented by a different metabolic pathway than when it is oxidized by respiration. Yeah, the answer is C. So A, B, C are both correct. Is that right? They have a different pathway. You have a hetero, homo, and when it's oxidized, it's all the same. How are the aerobic respiration and anaerobic respiration similar? So we talk about both utilizing organic molecule as an electron accept. The primary purpose of catabolism is what? We talk all day about anabolism, catabolism. What is that? What is the primary purpose? Generate ATP, is that right? Yeah. That's the primary purpose. Okay, again. In the cycling of sulfur compounds in the environment is involved in the oxidase H2S2 this. So you have to, this one you have to remember it. I don't think I taught. This is should be one of that is called Beggar talk. In our slides, which did not mention that, yeah, I don't think so. It should be these two, one of these. Okay, so this one you don't have to worry. 1 ton of Krebs cycle generates. You should be very familiar with this one right now. 1 substrate level phosphorylation is correct. 1 FADH and 3 NADH2. This does not include the transition step. So the answer is what? Is D. Okay? C is absolutely is wrong. B is wrong. They should have a substrate level. A is wrong because there's no FAD. So even you don't know, ABC is gone. Okay, how about the E? Of course there's no NADPH. This is usually in carbon cycle or antibodies. You're not going to be showing up in the carbon cycle. So the only thing left is D. Even you don't know, you can do the elimination of the others, you still can get the correct answer is D. It's very simple. You don't have to worry about this. I don't talk about chlorophyll anymore. This is the one is water. You don't have to do this. Okay? You don't have to do this. We don't talk. Okay. Which of the following is involved in generation of precursor metabolites? All of the above. You don't have to worry about this one. Okay, so that end up with all we have finished. Do the review of this section. So you should be very good. This is going to be your points, okay? Recite this. If you never read it, please go ahead and study. Not done yet. Okay. Who wants to come here volunteer to do some work, to the review for everybody else? Don't leave. I tell you, you're going to feel regret if you leave. I'm serious. Don't leave. Okay. We want to talk about that. Who can come here to the front? Who want to volunteer? Lacto opera. Who wants to volunteer? Okay. I'm going to draw, you're going to pick up one of the representatives, okay? P-O-C-A-P. Lac Z, lac Z, lac Y, lac A. Okay. Second, cap P O lac Z lac Y lac A. Cap P O lac Z lac Y and lac A. Cap lac Z lac Y and lac A. Okay, what are we going to talk? We had RNA polymerase. We had CAMP. There is a 4 different situations. Who wants to come? Any volunteer? Okay, yeah, good. Josh, Holland, is that right? Yeah, very good. You're going to explain. You can use notebooks. That's fine. So let's look at it. You're going to face them to talk.
All right.
So 4 situations. Explain them to me.
This first situation, lactose is going to be present.
Okay, lactose is there. Okay.
Yeah. Glucose is not going to be present.
Okay, no glucose.
And so
what's going to happen?
since there's not any glucose, CAMP is going to bind the cap.
Okay.
RNA polymerase will be, well, obviously it's going to be on the mRNA. And then the repressor that's on the operon here is going to be removed because
Okay.
lactose is going to come in and move that.
Okay.
So transcription will happen. And, yeah.
So we'll be using lactose.
Yes, lactose will be used. Second is there's glucose but no lactose. So that means that there's not going to be any CAMP.
Okay.
RNA polymerase will be attached to the promoter. And since there's no lactose to remove the inhibitor on, since there's no lactose to remove the inhibitor on the operon, RNA polymerase will not transcribe to the mRNA.
Okay.
And then here is both are present.
Okay.
So there's glucose and lactose.
That's what we're going to have to do. You were right there. Glucose is the lactose. Yeah.
So it's going to be CAMP. Wait, no, sorry. Not going to be any CAMP.
Okay, you can write on the side. It says it's not attached.
Yeah.
Just on the side. You can see it.
So it's not attached to cap
Yeah.
because glucose is present.
Yeah.
The suppressor will be removed
Okay.
because there is lactose present
Yeah.
and RNA polymerase. And you said it differently, but when I took 117, now Rotman was saying that it would transcribe very, very slowly,
Okay.
but you were saying that it did. I don't know. But yeah, no CMP, but the repressor will be removed.
Okay, you can see it's very slowly.
Yeah,
That's
just
a
how I remember it.
reason where we had a limited amount of the allolactose sometimes in the cell.
Yeah.
Okay, it's the same thing when this is small, slow. That's in the UK. What we call it? It's a human cell, but
Yeah.
you took it a human nutrition process.
Yeah.
But we said it's not going to transcription for bacteria.
Okay. And then there's no glucose,
Okay.
no lactose.
Okay. Then what's going to happen?
The inhibitor is still going to be here because lactose is there to remove it.
Okay.
And then I don't know for sure, but CAMP would be there because there's not any glucose to keep it from going.
Okay.
But there would not be any transcription because the repressor is still attached to the operon.
Okay. I would say about like 90% is correct. But there's one thing we have to mention is, okay, once this happens, this will combine. RNA polymerase and the promoter, they'll combine, which means they're going to start transcription. If they not combined, there's no transcription. So like this situation, of course it's not combined. It's just released. Okay? It's the first thing you need to do. So we want to measure again. If lactose is present and no glucose, what's going to happen? This cap will be attached to the CMP repressor will not attach to the operand, and then RNA polymerase and the promoter will be combined, so transcription will happen. Okay, that's important. The second is the second situation. If you have a glucose and a lactose will not be started to use. Remember, I said this is a very economic way for bacterial growth. So what happens? Once you have a lactose, repressor is not there. It's not in the O, it's released. RNA cap is not combined with CAP. So RNA polymerase will not combine with promoter. So this is key. Because they're not going to use. Remember, I said two of the person get together is not easy because this is positive and negative control has to be both works. No, the glucose and no lactose. The repressor will attach to the operand. And the glucose, this camp will not combine to the cap. Of course, RNA polymerase will not combine to the promoter. Nothing going to happen here. Once both are not happen, lactose, glucose, both are not there. Camp will combine to the CAP. And here, what you're going to see is a repressor will also attach to the O, so nothing will happen. Now, if you're still confusing, there's an easiest thing to do is, okay, I turn these off, is you look at your slides, the last one. Should we turn this off? The last one, obviously remember. The last slides of that examination 4, examination 3, slides number 4, is the one you should log. Is all the way at the end. Or not? It's wrong. Goes here. Stuff there. We go here. This is talk about translation and this is the slides what you should look at. Okay, if you're still confused, I think this explained it very well. The first situation, lactose, no glucose. Okay, the second situation, neither no glucose and no lactose. Lactose and the glucose both are there. And the glucose, but no lactose. Okay, so this is something we could be talking about that, and that could be the question, which is to ask you to fill the table. There are 4 different situations, so you have to explain, because lacto-operant is important. Okay, that's something I want to mention. Okay, I think we all finished here, and Thursday 11 o'clock I'll see you. I think I'll just skip it. I'm not going to go ahead to lecture, so afterwards we're going to talk about the food safety, poultry processing related to microbiology, and also immunology a little bit, and the final we talk about a virus a little bit. Okay.